Today we are going to do this question. Uh, this is from Cambridge Biology paper 4. Let's read the question. There are a number of mutations affecting the production of fetal hemoglobin, hemoglobin F, and normal adult hemoglobin, hemoglobin A. The hemoglobin A allele codes for the normal beta globin polypeptide of hemoglobin. Hemoglobin S allele, caused by a base substitution mutation, codes for an abnormal beta hemoglobin polypeptide. The base substitution results in the amino acid glutamine, which has a polar R group, to be replaced by valine, which has non-polar R group in the polypeptide. The abnormal hemoglobin molecules, hemoglobin S, form fibers in low partial pressure of oxygen. The fibers cause red blood cells to become sickle-shaped, and the cells can block blood capillaries. Individuals with adult hemoglobin molecules, there are all abnormal hemoglobin S have sickle cell anemia. This is painful chronic condition that can be life-threatening. Explain why this mutation caused the hemoglobin S to form fibers with two marks. Okay, first we will see what is the meaning of a mutation. We can consider mutation is the change of the base sequence of a DNA molecule. So this base sequence change is random, and the base sequence change may be single nucleotide change or maybe many nucleotide change. In substitution base substitution mutation, one base is replaced with another base. Example, let's assume this situation. Here there is a codon A, C, T. ACT is a normal DNA molecule, and ACT represents one amino acid. It codes for one amino acid. If mutation happen, A is still here, and C replaced with G. So here C substituted with G. This is called substitutional mutation, and this is called base substitution. Now here code for amino acid, let's assume X. In this case, it is not the X, it may be Y. So in sickle cell anemia, the glutamine replaced with valine, and glutamine has polar R group, but valine has non-polar R group. Okay, explain why this mutation caused the hemoglobin S to form fibers. This one is a hemoglobin normal, and this one is a hemoglobin S. Okay, when protein is synthesized, the first product is the polypeptide chain. The polypeptide chain is formed inside the ribosomes. The polypeptide chain is having the primary structure. The primary structure is the sequence of amino acid. So sequence of amino acid should be in the right order. In normal hemoglobin, glutamine is very important to have in this chain, and it is having the right amino acid. But glutamine is replaced here to valine. So it has a wrong amino acid. Then the primary structures start to fold. During the foldings, the R groups are very important. R group make different bonds to decide the tertiary structure. Between R groups, there may be four types of bonds: hydrogen bonds, disulfide bond, ionic bonds, and hydrophobic bonds. So it is important to make proper bond to decide the proper shape of the hemoglobin molecule. But when it is replaced with the nonpolar group, nonpolar R group in the valine, it is making a different bonds. When it make a different bonds, so this bond shape, the structure shape, so it is making a different shape of hemoglobin. So here the first case, this hemoglobin is a globular shape, this hemoglobin is a fibrous chain. Because valine has a nonpolar R group, it forms different bonds. This bond decides the shape of the hemoglobin and it produces hemoglobin S with a fibrous shape. Because bonds are formed between hemoglobin within hemoglobin S. Hemoglobin S, it forms these bonds. Question number 2, the B part of the question. Fetal hemoglobin, hemoglobin S is produced by the fetus until just before birth. Right? So usually the fetus has fetus hemoglobin, and after birth, then it start to produce adult hemoglobin. By the age of 6 months, adult hemoglobin has replaced most of the hemoglobin. Then 6 months, still fetus has hemoglobin F and hemoglobin A is increasing. So after 6 months, there may be no fetus hemoglobin and is replaced with more adult hemoglobin. These changes occur when the genes coding for hemoglobin F are switched off and the genes coding for adult hemoglobin are switched on. Another base substitution, British 198, causes fetal hemoglobin to continue to be produced. So if this British 198 mutation, there's another mutation, fetus is the adult after birth, fetus hemoglobin continuously produced. So normally age of 6 months, there is a 1% of fetus hemoglobin. But in mutation with British 198, we can find 20% of hemoglobin in adult. So adult has 20% of fetus hemoglobin. So this should be 80, this should be 20 then. Because of this mutation, adult is having large number of, which is 20% fetus hemoglobin. Hemoglobin F, fetus hemoglobin, has a higher affinity for oxygen at low partial pressure of oxygen than adult hemoglobin. Individuals who have both sickle cell anemia and British 198 mutation have reduced symptoms of sickle cell anemia. Now in this case, the person has both sickle cell anemia and also British 198 mutation. Suggest why having the British 198 mutation reduce the symptoms of sickle cell anemia. Okay, this person has hemoglobin S, hemoglobin F. Hemoglobin S causes sickle cell anemia. So hemoglobin S reduces the oxygen carrying capacity. Hemoglobin S forms sickle cell shaped. Hemoglobin S breaks the red blood cells and it can clog inside the capillaries. So it continuously forms the symptoms of sickle cell anemia. But having 20% of hemoglobin F, so it has the higher affinity for oxygen. So then the amount of oxygen carried by the person is higher than sickle cell person, because hemoglobin F has the advantage, so it can do better functions to take oxygen at low partial pressure of oxygen, and it transport higher amount of oxygen to the tissues. So this is the cause for reduced symptoms of sickle cell anemia when the person is having also British 198 mutation. In adult with the British 198 mutation, the gene coding for fetal hemoglobin polypeptide remains switched on. This is due to the presence of protein that control gene expression. State the term that is used to describe a protein that controls gene expression. Now, what is gene expression? Gene expression we can consider as the gene which is transcribed to the messenger RNA, and the messenger RNA translate into the protein. So first stage is a transcription, second stage is a translation. So production of a protein from a gene is called gene expression. So how this process is regulated? This process is regulated by different factors. In order to express this gene to messenger RNA, the enzyme required is called RNA polymerase. Now, RNA polymerase should bind to the special region, and the region is called promoter region. And this region is called gene. So before the gene, there is a special sequence, and this is called promoter region. If RNA polymerase wants to bind to this place should be facilitated by another group of proteins, and these proteins are called transcription factors. So transcription factors regulate the gene expression. Transcription factors can switch on, switch off the genes. In this case, RNA polymerase can transcribe gene into messenger RNA if correct transcription factors bind to the promoter region. So the answer for this one is transcription factors. Next, part C of the question, gel electrophoresis can be carried out to test individual for the different version of hemoglobin. Hemoglobin A, hemoglobin S, hemoglobin F. A buffer with alkaline pH is used to make all hemoglobin molecules negatively charged. Hemoglobin S molecule have an additional positive charge compared to hemoglobin A. Describe and explain how gel electrophoresis is used to diagnose sickle cell anemia. Okay, hemoglobin S has additional positive charge. Right, in gel electrophoresis, we place these proteins in different lanes. Let's assume this one is hemoglobin A, hemoglobin S, hemoglobin F. On the starting side, negatively charged, this side is positive. This is a negative end, this is a positive end. This side is the cathode, this side of the anode. So when the gel is placed in the buffer solutions and applying the electrical current, then the DNA molecules, protein molecules moving to this direction. Negative end to positive end. The distance moved by this protein molecule are decided by the charge and the size of the molecule. So in this case, we don't have a clue about the size of the size of the protein molecules. If we assume the molecular size is same, only here the hemoglobin S has positive charge. So hemoglobin S can run less distance because it's having positive charge. Its moving is slow because it cannot move very nicely, comfortably from positive to positive end. So hemoglobin S distance travel is very less, but hemoglobin A and hemoglobin F can travel. So if we have a reference band here, and we can find hemoglobin S, hemoglobin A, and hemoglobin F. So hemoglobin S travel the lowest distance, but A and hemoglobin F travel the longer distance. Based on that, by comparing the bands and by comparing the band's position with the reference band, we can decide which one is the hemoglobin S. Four individuals had their hemoglobin analyzed by gel electrophoresis. One of the individuals was heterozygous for hemoglobin A and hemoglobin S allele and had a condition known as sickle cell trait. So the person has hemoglobin A and hemoglobin S. So normal phenotype, we should expect hemoglobin A, hemoglobin A. 1 month old baby with normal phenotype. So we have learned baby continues to produce hemoglobin F until 6 months. So then we can have hemoglobin F and hemoglobin A. Now in this question, we are advised to use this combination, hemoglobin A and hemoglobin S, because it's a sickle cell trait. Sickle cell anemia should have hemoglobin S, hemoglobin S. So here two different bands, then these two different bands travel. So hemoglobin A reference band is here. Here is hemoglobin A and we can place the band here. And hemoglobin F reference band is also very close to each other and we can place hemoglobin F here. In this case, there is only one band because both are hemoglobin S, so we can see one band here. Now, this is hemoglobin A and S. We can see two bands, hemoglobin A and S, hemoglobin A and hemoglobin S, because it's a sickle cell trait. And lane 4, individual with sickle cell anemia has hemoglobin S and hemoglobin S. There it's one band. So hemoglobin S only. Here is a hemoglobin A, hemoglobin S. Here hemoglobin A, hemoglobin F.